All right, it's time to begin again in our international portion of the ISSR&D conference technical sessions. This session is International Biological Research on the ISS. Our chairman for today for this session is Peter Batenburg. Peter is from the Netherlands and he is from Hqspace. So Peter, thank you for coming in this afternoon to you almost. This session today contains 5 presentations, and we'll see them on your screen momentarily. So lots of interesting things there in the biological worlds, and it's amazing to understand some of the things that are going on around the world as we do these different elements of the ISSR&D conference. So now we're want to remind you to please go to the bottom of your screen and click Q&A and enter your questions for the Q&A. Don't use chat, use the Q&A function, and we'll remind you again about that in a little while. That's really important to us. We want to get your questions so that our presenters are able to respond almost directly to you. So please do questions and answers. So now we have 5-minute executive summaries of each of these briefings, and we'll run those from beginning to end with as few stops as we can, only ones where we have to stop it momentarily to move forward. And so Sarah and Patrick, please run our slides.
Welcome everybody to the lecture on human osteogenic differentiation in space, molecular clues from the SERiSM project, of which I am the principal investigator. This study was performed by University Campus Bio-Medico Rome in collaboration with Tor Vergata University of Rome and with the technical support of Kaiser Italia and the University of Teramo. As you see, SERiSM is an acronym where the words endocannabinoid system are included. And I have to recall that endocannabinoid system is indeed a complex array of lipid signals called anandamide AEA and 2-arachidonyl glycerol 2-AG that are able to activate G protein coupled receptors like CB1 and CB2, cannabinoid receptor type 1 and type 2 on the plasma membrane, and others, and most notably able to be kept under strict control by metabolic enzymes that can biosynthesize endocannabinoids like NAPE-PLD or Degl'Innocenti, and degrade them like FAAH and MAG lipase. Signaling system is very important in many pathophysiological circumstances of our body, and of relevance here, they have been recognized as drivers of bone remodeling, acting both via CB1. You see they reduce the sympathetic tone, the endochondrial bone formation, and by CB2, where they increase age-related bone loss and bone turnover, and proliferation of osteoblast progenitors. They are so relevant for bone remodeling that genetic ablation of one cannabinoid receptor is enough to severely impair the trabecular architecture of mouse bones, as you see on the right of this slide. And on this basis, we thought to investigate whether via endocannabinoid signaling, blood cells taken from peripheral blood of our body and deprogrammed and then re-instructed to make bone material, these cells could be a way to compensate or even cure osteoporosis observed in the astronauts and possibly also on Earth. The idea is that you can re-inject these blood cells educated, driven towards the osteogenic differentiation to make bone material, and when you re-inject them in the same donor, you can help him, the donor himself, to cope with the disease. Our experiment, the SERiSM, was flown in August 2017 on board the SpaceX CRS-12 and was launched to the ISS. There we could measure imbalance of the endocannabinoid system elements, showing that CB1 signaling was enhanced while CB2 signaling was reduced, and also that degradative enzymes like Fa and MAG lipase were reduced, leading to increased levels of endocannabinoids. Showed that protein expression of transcription factors that are critical along the osteogenic differentiation were modulated in such a way that osteogenesis could take place to a larger and faster extent in space compared to Earth. Indeed, we found increased SOX17 and GATA4 that are pro-osteogenic transcription factors, and decrease the OTX2 and snail that are kind of repressors of this differentiation in space compared to Earth. So in low gravity LG compared to ground gravity. Our results have been reported in a few papers, and I refer the people interested in this topic to scientific reports in 2019 and AMS in 2020. Thank you for your kind attention.
I'm Andrea Degl'Innocenti, and today I will talk about antioxidants for space using nanotechnology and a simple. Space causes different sorts of stress, both on astronauts and their crops, with a common component of oxidative stress. And our main focus is going to be the brain, which poses particular challenges for space. The solution we propose are planarian worms, which are simple animals, already a model for degenerations, aging, and stem cell research. They are easy to grow and they are prone to regeneration. Indeed, they may allow for a timely control over brain development, at the same time providing a low-tech platform for genetic studies, even directly aboard the ISS. In brief, could planarians be an ideal model for space? Apart from that, we aim to test the development of a full head directly in space using their regeneration capabilities. And also, we would like to study oxidative stress and to counteract it with technical antioxidants. We know that space induces oxidative stress, and therefore people have been attempting to use organic radical scavengers, but these are perishable and they tend to be consumed as they are used for their biological function. Instead, Nanoceria or cerium nanoparticles might be a self-renewable and long-lasting ROS scavenger, which might turn useful for long space travel. Our study is actually made by 3 different projects. 2 of them study Nanoceria in artificially altered gravity. This is where we are now, basically. The third one is currently a proposal, which is called the Simple Plan. Its aim is to study omics of planarians directly aboard the ISS, both on intact or specimens. About the basic characterization we already performed, we asked ourselves whether Nanoceria could enter the planarian body, and the answer is yes, by transmission electron microscopy. To understand the effect of artificial hypergravity and Nanoceria, we first performed the regeneration studies that showed that hypergravity hampers regeneration, with Nanoceria that seems to rescue this effect. And with immune assays, we saw that hypergravity increases the cell determination rate with some rescue from Nanoceria. Finally, artificial hypergravity decreases the mitosis rate. Here is a sample of our bulk transcriptomics data. In particular, here you see a Venn diagram showing differentially expressed genes for Nanoceria. Back to the outline of our project, let's see its third chapter. To study the effects of altered gravity and space radiations, we would perform an experiment with knockdown or normal animals with or without antioxidant directly in space, even on regenerating animals, and then do the same procedure on the ground. We would analyze samples as soon as we get them from space. Full experimental design would exploit the proposed aquatic habitat systems, but we could also do with a quick incubator, still producing available. In general, we believe our experimental design is tunable to fit all available room. A third stressor is variations on environmental pressure, especially hypobaria, which we think might characterize environments such as space greenhouses, and we would study that using the Antarctic Concordia research. This is our team, and you also see a list of previously funded space projects, in particular with two ISS-related projects detailed in Abstract 304. Thanks for your attention.
Good morning or good afternoon. My name is Rosa Santomartino, and I am a researcher at the University of Edinburgh in UK. In this talk, I am going to discuss the potential roles of microbe-mineral interaction for human space exploration, with particular mention to biomining. I will show you the results from the ESA BioRock space experiment performed on the International Space Station in 2019, and briefly tell you something about the future space experiment we are about to perform, BioAsteroid. Microorganisms such as bacteria and fungi perform countless tasks on Earth and are necessary for human survival. While being important on Earth, they will be absolutely essential for human survival beyond Earth. As we move into space, we can harness microbes to make our lives easier and improve the success of space settlements. Biomining is the process of using microorganisms to extract metals of economic interest from rock ores or mine waste. Space biomining represents an excellent way to obtain needed materials on other planetary bodies, rather than relying on continuous resupply from Earth. However, to understand if we can perform biomining in space, we first need to test how space conditions, especially microgravity and partial gravities, influence microbial interaction with rock and mineral material. It is with this idea in mind that we performed the first biomining experiment on a space station, BioRock, that was launched on the International Space Station in 2019. We selected 3 bacterial species for this experiment: Sphingomonas dissecabilis, Bacillus subtilis, and Cupriavidus metallidurans. Sample preparation consisted in the inoculation of a basal slide, chosen for its natural porosity and because it is considered a good analog for both lunar and Martian rocks, with a single bacterial culture, while negative controls were sterile basal slides. After preparation, each sample was integrated into the hardware, the biomining reactor or BMR, that was specifically developed by Casa Italia for BioRock. A separate set of samples, including triplicates, was subjected to microgravity, simulated Martian gravity, or simulated terrestrial gravity on the International Space Station. A ground control was also performed, and these samples were subjected to real terrestrial gravity. Coming now to the results, what did we discover from the BioRock experiment? We first analyzed the final cell concentration reached by measuring both direct cell counting and optical densities of the liquid cultures. The results showed no difference in final cell concentration between the space samples after 21 days of growth. This indicates that, regardless of how the different gravity regimens may have influenced the bacterial growth over time, after 21 days, They all reach the same final concentration, indicating that low gravity is not a limit to space biotechnological applications involving microorganisms on Moon, Mars, or other celestial bodies. Analyzing the samples with mass spectrometry, we also discovered that one of the three organisms, Sphingomonas dissecabilis, was able to effectively biomine rare earth elements in all the gravity conditions tested. Results in microgravity were not statistically significant. However, this is probably due to a contamination occurred in one of the negative controls in microgravity. This important result provides the first demonstration of biomining on a space station under a range of gravity conditions. We are now preparing for a second space biomining experiment called BioAsteroid that will be launched on the International Space Station at the end of 2020 and that will use again the biomining reactor. This time we will investigate biomining of real asteroidal material in microgravity. In conclusion, with BioRock we provided the first demonstration of biomining on a space station and showed the feasibility of space bioindustrial application involving microorganisms under a range of gravities. We demonstrated the functionality of a novel space biomining hardware and discovered a novel biomining microorganism. There is a lot yet to be understood and research is needed. I would like to thank the whole excellent BioRock team and all their components, and you all for your kind attention.
Thanks to all the viewers. I am Lakshmi presenting under Plant Hacks on Biomimetic model of plant vascular structures for extraterrestrial impact study and I would like to give an overview of digital twin for plant and brief out various factors involved in modeling. So, what actually is the need to study plant beyond biology? The physiological process of transport of water and minerals, sugar translocation, transpiration rates are still requiring validation for various theories. These experiments require a precise incubation, sensor tracking and monitoring setup at ISS. So, prior to this expedition, we would model the plant structure and processes, simulate various theories and assumptions under microgravity or other extraterrestrial biome conditions, which also helps to design the experiments effectively. This interdisciplinary project with science and engineering translation under single platform for designing biological structures as geometries, modeling the physiological process as mathematical and experimental physics study is motivating. Coming to gravitropism, many past theories presented the gravity as major factor in various traits of plant growth and functioning which are not yet tested. The experiment done at ISS had questioned the Darwin's theory of root growth direction with respect to gravity, since the growth proved that it is beyond the gravity. It's time we know the actual involvement of gravity on plant functionalities. Mechanical modeling of plant structures with mathematical profiles can be done, but to use the current 3D scanning and other imaging techniques to obtain the actual plant structures with precision and to model the complete topology. Data on plant structures with respect to phenotypes for various species is available, which can be modeled using 3D reconstruction methods available currently with computer vision and deep learning tools. The primary focus xylem structures are lignified cellulose vessels, with cellulose being hydrophilic for adhesive nature of transport. The material is created with properties and is applied to the geometries to make the model further effective. Since the physiology is highly dependent on external environments, just as we test the antennas and other electromagnetic devices in simulation radiation box, the atmospheric parameters which are available so far like precipitation, humidity, gases density, CO2 concentration, etc. for Earth and other sources are deployed as material to bounding box with XML files specifying the parameters. Focused on modeling the plant structures and radial flow, basic fluid analysis were only simulated. By modeling pipe flow and laminar two-phase fluid flow on xylem vessel extracted, the CT theory, Poisson-Lees flow, Murray's laws, etc. are modeled for pressure and tension-driven fluid flows. Aldis and Simpson models are modeled for inter-vessel pore transport and osmotic modeling. The matrix of pressures, potential, velocities, osmotic potential are modeled for any given point on the simulated plant to obtain spatial and temporal references. Of offers not only experimenting and validation on theories but also provide distribution profiles of various parameters with respect to gravity and atmosphere. To study the hydraulics of plant under microgravity conditions with limited atmosphere. Will it be transpiration driven as on Earth or capillary driven? A new hypothesis may be predicted. Also, calculation of energy budgets pressure profile can help in creation of life support systems for extraterrestrial farming. A vision for standalone plant with artificial root implanted to compensate atmospheric CO2 and other gravity deficits for non-terrestrial locations can be tested with this digital plant. The vulnerability curves which define the boundary and critical parameters and conditions of plant in various environments can be obtained not only for extraterrestrial farming but also for terrestrial practices of agriculture management. The model mentioned offers a great scope for adopting new farming practices even on Earth. This is the model being prototyped for infusing the fertilizers, insecticides and any solutions of certain ranges of viscosity directly into the tree vascular system in correlation with tree susceptibility to pressure and dosages. We have modeled the fertilizer distribution with respect to actuation provided by this model of simulations. The implant to tree as artificial root with micro actuation and channel setup as plugin for tree any number of times for effective tree management, which offers zero environmental effects replacing the soil mixing and spray methods. With inspiration from various experiments conducted in microgravity environments and understanding the scope it offers, not only for extraterrestrial but also farming techniques on Earth, we vision to create a complete digital plant for various species. Thank you.
Bioreactor Express commercial service to perform experiments in microgravity. A status after 1 year of activity. In 2019, Kaiser Italia partnered with the European Space Agency to set up Bioreactor Express, a service conceived to make access to microgravity a reality for companies, universities, and research organizations wishing to perform scientific experiments and technology demonstrations in space. The partnership was made possible by 3 concurrent factors: the availability of low Earth orbit resources and the commitment by the European Space Agency to boost their commercialization, the know-how gained by Kaiser Italia in the development and implementation of experiments in microgravity, the existence of potential market for microgravity experimentation. So far, 3 experiments have been acquired and are in the pipeline. In all 3 cases, the experiment is performed using existing hardware with flight heritage. That cuts down the design and development time and reduces the time necessary to certify the flight readiness, both from mission success and safety point of view. Experiment 1 is a microbiology experiment commissioned by a university to investigate how gravity affects the interaction between microbes and rock in reduced gravity. In this case, the principal investigator is well aware of the space environment, because he already performed a similar experiment in space in the recent past, using the ESA institutional approach. In fact, he used Bioreactor Express to get access to space in a short time to continue his research. The experiment is scheduled for launch in October 2020. Experiment 2 is commissioned by a large company supported by a university. The experiment is part of a commercial research to develop novel therapeutic nanotools to target central nervous system diseases and cutaneous cancers such as melanoma. In this case, both the company and the university involved in the experimentation are new to the space environment, although experiments in the same field have been already performed by others on the ISS. The experiment is scheduled to launch in 2021. Experiment 3 is commissioned by a startup supported by a university. The experiment is part of a commercial research to develop bone substitute for bone grafting, a procedure of skeletal reconstructive surgery. In this case, the startup involved is new to the space environment, while the university is well aware of it. The experiment focuses on the microgravity-induced effect of osteoporosis, which has been a target of several experiments performed on the ISS so far. In order to show what sort of experimentation is possible on the ISS, Kaiser Italia established the Hqspace, a database of technical and scientific publications that aims to collect the knowledge and capabilities developed with space experiments performed on the International Space Station and other low Earth orbit platforms. Hqspace provides general information on the experiments performed and allows the navigation to the sites hosting the publications, through selection of payloads, investigations, or scientific categories of interest. The database is updated with the latest known publications. This database complements the ESA Erasmus Experiment Archive and the NASA Space Station database. For any further information, you can check out our websites bioreactorexpress.space and kaiser.it. Thank you for.
All right, so we have a lot of different, fairly highly technical briefings here. And I want to make sure that you guys recognize that we do have the Q&A function. We have a few questions in. The Q&A button is on the bottom of your screen there. You see a arrow pointing towards it. Just type in either your presenter or your presentation name that you want to give it to. Just one or two words of the presentation title will work. And then give your question. Peter and I worked together in the IAF, and he and I were having a little roundabout yesterday, trying to say, well, if we need some additional questions, let's get some questions from you, Alan. And I went through and created a few questions. I did not send them to him because I realized they were absolutely horribly naive. So I would really like to invite the audience to find some significant questions that can give this great panel. So Peter, you've got it.
All right, thank you, Alan, and welcome everyone to this session on international biological research on the ISS. A warm welcome to our panelists. We have Rosa, Nandamuri, Mauro, Andrea, and Alessandro here today. So thank you for coming in. Thank you very much for your very interesting presentations. So I'll start off with a question that we got from Muntasir Ali. That's a question to Rosa. He compliments you on the presentation, and he asks, do you know the radiation sensitivity of the microbes used in the BioRock experiment?
Yeah, thank you for the question. So basically, while we know a little bit about Bacillus subtilis and Cupriavidus metallidurans because they have been widely studied, for Sphingomonas we don't have as many information because there are not many studies on it. However, we are of course studying it. And the thing that would be very interesting would have been to analyze like some omic approaches like transcriptomic or genomic on the sample when they came back on Earth. But unfortunately, with the fixative that we choose for some technical reasons, we could not retrieve DNA and RNA. So unfortunately, we don't have any data from this point of view. But of course, what we are doing now is trying to sequence the genome of Sphingomonas and trying to understand what happens a little bit in the from the transcriptomic point of view, so that we can try to understand what happens if they are resistant to radiation or not. Again, from the point of view of Bacillus, Bacillus is highly resistant to a lot of harsh environments such as space condition and high radiation. So we know that he is resistant, and Cupriavidus as well has his strategies to do that.
All right, thank you. Muntasir, I hope that answers your question. If it didn't, then please feel free to resubmit another question in your comment box. Ellen, you mentioned you prepared some questions, so if you want to ask one, just let me know. The floor is yours. Let's see if Alan comes back online. There he is.
I was taking my nap. So yes, talking about the BioRock. The last slide showed a tremendous number of collaborators or a tremendous number of people on your team. So help us understand a little bit about what you do yourself as BioRock versus what the other teammates do, how you manage that huge activity.
Well, I was quite lucky because the whole team, we had a very good collaboration. So we really collaborated in the real sense of the term. So I was responsible to lead the science team together, of course, with my supervisor, there is Professor Coquel. But the BioRock actually started, the project itself started in 2008. So I only came into the project in 2018, and I basically made it I made all the preparation until the launch, and then I analyzed the samples when they came back. And part of the science team was also DLR from Germany, the German Aerospace Center, and SEKSN from Belgium. Then there was, of course, Kaiser Italia, who's, you can see Alessandro Mariani is from there, and he collaborated with us for the creation of the hardware. Actually, they created the hardware with our collaboration, and then ESA, of course, and then there was all the people from Aztec. It was a very wide collaboration, but to be honest, it was not complicated as one would imagine, because everybody had, we were very collaborative with one each other. And in terms of what I did, again, I prepared the whole experiment and did all the analysis, the following analysis, and I'm writing and wrote the papers that have been published now or are under review now.
All right, thank you. Ellen, I hope that answered your question. Thumbs up, so that looks good. So I have another question coming from Carolina Marin de Viscova to Andrea Degl'Innocenti. She's asking what species of planaria were studied or will be studied.
Okay, hi all. Thanks, Carolina, for the question. Basically, the whole project is based on the planarian species Lugesia japonica. And in our experience, is the best taxon for such a study because it is very easy to handle and rear, and also it is well apt for toxicological studies. Another option would have been Smithia mediterranea, which is also a popular model for stem cell research. But I mean, we are open to that. For now, we are focusing on Lugesia japonica.
Okay, Carolina, if you have a follow-up question, please let us know with the Q&A. And in the meantime, I have a question for myself, for Nandamuri. You outlined your work on modeling the stream of fluids within plants and the purposes that benefits that we can get from it, from modeling that, and also adapting it for how it would work on the moon or Mars or on other occasions. Based on your work so far, do you want to proceed with tests or experiments on both the ISS to validate your models? What are your thoughts on that so far? Can you unmute yourself?
Actually, we are currently in the modeling phase. We are yet to test various theories of like radial and axial flows. So once we get any lead, like it may be something disruptive, or it may be some surprising results can be expected, then we would be going to flight on ISS. Actually, already there is a plant habitat system at ISS to experiment readily. With the help of the farmer like live coding, we could do the experiments live with the available facilities itself. So we are looking for something which may surprise or such setup.
All right, thank you. Then I have a question to Mr. Mauro Maccarrone, if that's my correct pronunciation. Based on the research that you performed with SERiSM on board of the space station, did you get any, let's say, do the results stimulate further tests on ground, maybe also for medicinal therapy for people on Earth suffering from osteoporosis?
Yeah, thanks for your question, Peter. We believe that we are just at the beginning of something that might hold promise for therapy on a number of bone pathologies on Earth. The fact is that we found a new way of addressing the problem by using peripheral blood stem cells, not mesenchymal cells that are more commonly used to address the problem. And if the concept of an autologous treatment of the subject with his own blood cells re-educated to become osteoblasts works on Earth, then we have something totally innovative. The experiments in space show that, in fact, this can happen. It appears to happen faster in space than on Earth, but we have a model that we can still exploit on Earth. We know how to deprogram and reprogram, once again, autologous blood cells. And you know that nowadays, the idea of taking blood from a patient, modified somehow, and re-inject the same blood to that patient is absolutely a routine in so many diseases like kidney disease. So we think that we have something to be further exploited, no longer or not only from a preclinical, experimental, basic science point of view, but also for a real therapeutic application.
I think that is a very impressive work you're doing, and it's one of the things where you see what the benefits of research on the ISS directly benefits human lives in a very direct way. So, very impressive research.
Thank you.
In the meantime, I received a follow-up question from Carolina to Andrea. Will your studies focus on the stem cells, the neoblast, from the effects of radiation and microgravity? And then how do you separate the microgravity and radiation in your studies?
Okay, so about the first question, I would say that stem cells, the neoblasts, are not the main focuses of our work. I mean, not directly, but still they are, of course, a very interesting target for such a strand of research. And therefore, we are evaluating currently single cell omics experiments that, especially if combined with fax sorting, could yield precious information about neoblast point. So telling apart radiation effects and microgravity, I have to say that up to now we have focused on microgravity and hypergravity actually alone. But in the future, we actually plan to study radiations as well. And the idea is to tell these two factors apart by exploiting systems that give the opportunity to use a centrifuge in space, so that specimens would be exposed to radiations while having an Earth-like experiencing an Earth-like gravity. So this is more or less the goal. And another thing, of course, when you have such an experimental design, you can also attempt to study radiations alone by subtraction in basically Venn diagrams in transcriptomic datasets.
Okay, Andrea, thank you for your answer, and Monsieur, I hope that answers your questions. Again, if not, let us know. In the meantime, I have a question for Alessandro. You showed us what you've accomplished with the fast track of experiments for the space station. This is, I think, something that's been very desired in the scientific community, because the lead times to get something into space tends to be maybe sometimes too long for the researchers within a research period. And you've given a couple of examples, and I think a very nice one is that we actually have one of the scientists here in this panel with Rosa. Can you enlighten us a little bit on how the advantage of the fast track, or if there is a lot of compromises that go into reutilizing existing hardware?
Hi, thanks. Can you hear me?
Yes. Yes.
Okay.
Go ahead.
Hi. Thanks for the question. Yes, indeed, the fast track, which is roughly 12 months from, let's say, experiment definition to the actual launch and the execution of the experiment in space, is possible in the case you have already the hardware ready. So either it is someone who has developed some hardware that can be used and has all the certifications needed to fly, or it is something that we already developed in the past, and we know how it works. We have the certifications, the tests, and so far. So that is one point that needs to be considered. The thing is that Kaiser Italia has been doing this task, this job for I think it's roughly 2 decades now. So we have rather a large portfolio of experiment hardware that covers different biological models, and that can be used as it is, or can be customized in a way that still allows to get to the flight in a short time, and makes it possible to incorporate possible other requirements that are not already in the hardware. So that point is correct. The fast track is true, but you have to use the hardware that has been developed. On the other side, what makes it possible is that you have a single point of contact between the scientist, let's say, or the researcher that has to perform the experiment and Kaiser Italia. On the other side, you have the European Space Agencies, but between Kaiser Italia and the European Space Agency, we already have an agreement in place with pre-allocation of resources and a sort of streamlining of all the activities that need to be performed. So this is the way we can make a space experiment in a short time, and also with a cost that is much less than it would be with the institutional approach.
Thank you. And Rosa, can you maybe tell us what that means for you? Because I think it's your experiment that has a very fast follow-up. So what does it mean for your research?
Oh yeah, so it was, me personally, I felt much more like confident on what I was doing, because I already knew anything. And also now that we are preparing for BioAsteroid, that is, as Alessandro said, is going to be launched in November, basically, hopefully. Everything is much more smoother. Of course, you still need to learn the science behind as a researcher, as a scientist, but I guess in my case, I was kind of more fortunate because I was really using exactly the same hardware that I was using for BioRock. So it was really, really easy. But I think the experience is very, very positive. I will recommend that if I can, because much faster. I am a postdoc, so my grant is for 3 years. And this gave me basically the possibility of work on 2 space experiments in the range of my postdoc instead of 1. So very good from my point of view. So yeah, positive experience.
I think Alessandro, that shows how happy you can make researchers. So that's, I think, something fantastic you're achieving. So one follow-up up to that. So your experiment focus now on the experiment that can go into the cubic facility. Are you also looking into other facilities on board the space station for which you can, let's say, propose this fast-track experiment payloads?
Okay, the agreement we have in place is for cubic. However, we already are trying to extend this to similar other facilities, custom facilities that we are developing, and also something that is being developed by others. The thing is that when you have the so-called the bioreactor, the outer interface can be you can build a facility that easily adapts to that outer interface. So you can have the same bioreactor performing the task in cubic or in any other facilities. The answer is yes. We are trying to extend the bioreactor express also to other ways to perform the experiment that are not cubic.
All right, wonderful. Thank you. I think it's again an opener for many to do research on the station. I have a question from Muntasir Ali for Nandamuri. Question number 1: is there any specific plant species that you are interested in using currently to building the model?
Currently, we have data on wheat plant and grapevine. And also various imaginary data and 3D data is available. So we are constructing the entire, reconstructing the entire plant in order to design entire radial and axial flows. And there is a Yale experiment which is done using X-ray machines to get entire and root anatomy. So these are used for individual element studies. Currently, we are focusing on wheat and grape. Yeah.
Okay, thank you. The second question is, SEP flow measurement systems are already available commercially. Do you have any plan to use these first in a lab experiment before sending them to the International Space Station?
These SAP flow experiments and all are using like these gogs and centrifuges in order to measure the xylem flow and hydraulic conductance, which prove there is a lot of difference between the measured value and the values when actually running in the plant. We would like, which is differing from several factors as the size of plant is increasing. So we would like to give exact SAP flow measurements within the tree without any with more efficient and accuracy. And we would like to, definitely. Actually, we are looking on the data of the current ISS project, APH, Advanced Plant Habitat, with which we are modeling the transpiration rates and all the environmental conditions based on how the real plant has given. So we would like to first create an exact environment and better simulation conditions, and then we will look forward to various species of plant.
All right, thank you. And Muntasar has a couple of more questions. So first of all, Muntasar, thank you for all the questions. These two are for Rosa. So question number one, is there any plan to use other microbes apart from the ones shown in your presentation?
Yeah, so not in this moment, to be honest, but so of course, when you have a space experiment, you have, of course, you want an organism that can bioleach, but you also have to accomplish other, these organisms that you use in a space experiment need to have also other characteristics. Like, for instance, in our case, it was survival to desiccation, survival to, for instance, to radiation, or not pathogenic. I mean, the requirements for microorganisms can be not only restricted to the thing that you are studying, but wider. So this is the reason why we selected the 3 organisms, and now we also selected the fungus for BioAsteroid. So we are not planning to use any other bacterium or fungus or microorganisms in general now, but the study is always on to try to find the best bioleaching microorganisms. And the best bioleaching organisms on Earth, microorganisms, are actually organisms that actually like very harsh environments, such as very, very low pH. That is basically is probably not the best option for a space experiment. That's why we choose these organisms that like neutral pH. But you never know, maybe in the future you can develop a hardware that will provide the possibility to study organisms that can be used with a lower pH or higher pH. So yeah, the search is on. Not only from my group, but I mean, there are other groups, especially in US, I think, that are interested in studying space biomining.
All right, thank you. And the second question he was asking is, what is the reason for using fungus together with bacteria for the biomining project?
This is a little related to the first one, and I will get to why. So the reason to use the two together, 2 microorganisms together, is that when they are in nature, they are not isolated microorganisms. Normally, you never find a single microorganism, but normally you find a community. So the idea behind was to try and see if you put the 2 microorganisms together, are they going to compete, or are they going to bioleach better when they are together? But still, it is still just 2 microorganisms, so it has the advantages of still being able to distinguish the 2 and not get the results too complicated for the analysis. And this gets to the first question, because I think one very interesting opportunity will be to study, to collect like a microbial community that can bioleach, and try to grow that on the International Space Station, and try to perform a biomining experiment on a community. Although, again, it would be maybe harder from the point of view of the analysis, but still very, very interesting.
All right, thank you. I have a question for Mauro. Can you tell us on what would be your next steps in your research, in particular, maybe for a next experiment to be performed on board the ISS?
This is a good question, and partly covered by some confidentiality. Basically, what we would like to investigate further are mechanisms behind the bone remodeling. And certainly, we still want to look at lipid signals. If you had the chance to look at my full presentation, you might have seen that we have a little history, a little background in lipid signaling in space. And there are new mediators that are very much involved in the resolution of inflammatory processes. And these new mediators might deserve some attention also in space. So at this point, I can only say that we have made a proposal to NASA for a new project to be launched on board the ISS. And if successful, we will add a little piece to this puzzle of how lipid signals can drive key events like bone remodeling. And a little extra I can add is how immune cells might contribute to that.
Sounds interesting. So looking forward to that, and thank you for sharing something, although I can understand that you cannot share all the details. I have another question from Alan. Alan is asking a question to Alessandro. So, sorry, I'm mixing things up here, Alan. I have another question from Alan first for Biometric. Or Alan, did I misunderstood that you also had a question for Alessandro?
Question on the Bioreactor Express, and he typed in the answer, but I think it'd be nice to give it verbally so everybody can understand it. What's your timelines from the time you get a contract from a customer? I assume you're primarily an implementation partner type person, or you provide the facilities. What's your timeline from contract with a customer to flight to recovery of data?
The timeline is 12 months from the acquisition of the contract to the flight, in case existing hardware is used. The recovery of the data, of course, depends on the mission duration, and of course the analysis is performed by the scientists.
All right, thank you. And microfluids are really interesting, and I'm really surprised at the amount of stuff that's being done with microfluids. So biometric microfluids, with all of the different companies that are beginning to do microfluids, are we beginning to see some competition for customers out there, or competitions for funding between the different companies?
Yes, indeed, there is some competition starting, although the market is very at early stage. I mean, I'm not complaining because luckily we are basically on schedule with respect to our expectations. But I mean, there is potentially a market which is much bigger than what we seeing and seeing now. The competition is there, although we target different facilities probably, and I expect that at least for a while. Sorry, let me put this way. It will take a while until we can see a real competition in the sense that we know here on Earth.
All right, thank you. We're approaching the end of our time here. I have one question for Andrea. You mentioned the 3 variables in your research, which is microgravity, radiation, but also pressure. And you mentioned that you want to do an experiment for the hyperbarrier effect, and you want to do this on the Concordia South Pole Station. So I was interested to see why the South Pole Station. What is it particularly offered to do your research over at the South Pole?
Okay, thanks for the question. It's actually a nice one. So the idea is to use 3 batches of planarians. 2 of them we would put at our facilities in Italy, and the third one actually would overwinter at Concordia station. And I think this would offer us a kind of unique occasion to study chronic hypobaria there, with a very low-tech approach, and also at the same time a very effective approach. I said that we would study 3 batches because one of the 2 batches that we would keep at our labs, we would then bring with us in the Antarctic summer in order to study, in that case, acute hypobaria. So I think we would have like two birds, one stone. And there we would perform behavioral studies and also regeneration, because we could regenerate without problems. But there at Antarctica, we would see the effects of both chronic and acute hypobaria on that. Also, another thing which is interesting to study is behavior, because we know that especially long-term hypobaria causes cognitive impairment. And so we would be able to quantify that, which is also an important aspect of the central nervous system.
Peter, I think we're out of time. Thank you, sir.
I think we are. We lost you a little bit there at the end, but I want to thank all the panelists for their excellent presentations and the excellent answers to the questions. All of you, good luck with your following research and your following work. Magnificent work you've done around there. So enjoy the rest of the conference, and I hope to see you on the next edition. And Ellen, the floor is back to you. Thank you very much.
Thank you, Peter. I believe this has probably been the most technical session that we've had so far, so that's really good. And I even understood a little bit of it, which was fun for me. So now we're going to take a break. We are going to come back with our next session, which is Human Health in Space, Part 1. We'll be back at 1:15 UTC. So that's about Eastern Time is 4 hours behind that. So 1 p.m. UTC is our, I'm sorry, 1:30 p.m. UTC is when we come back, 1:30 for Human Health in Space 1. And thank you all very much. We'll be back in 15 minutes or so.